Hi, my name is John. I'm a second-year MD PhD student in the Longer Lab, and it's my pleasure today to present this poster entitled Quantitative Analysis of the Collagen Matrix Ultrastructure after Myocardial Infarction. The motivation of this research is that cardiac fibrosis is the key hallmark of nearly all cardiac diseases. For instance, atrial fibrillation is characterized by fibrosis of the atria, and myocardial infarction leads to fibrosis of the ventricles. So what is fibrosis? Fibrosis is characterized by excessive extracellular matrix deposition after injury. The accumulation of extracellular matrix after injury is a normal response. For instance, after a myocardial infarction, otherwise known as a heart attack, cardiac fibroblasts create a dense collagen scar that replaces the dead cardiac muscle. And this prevents acute cardiac rupture, which would otherwise lead to, as you might expect, cardiac tamponade and death. Over time, however, the fibrotic scar becomes maladaptive, and it triggers hypertrophy and fibrosis of the remote myocardium, eventually leading to ischemic cardiomyopathy and congestive heart failure. Despite the central role of the MI scar in the pathogenesis of ischemic cardiomyopathy, the formation and remodeling of the scar over time is poorly understood. So in this study, to better understand the MI scar formation, we sought to quantitatively analyze the changes in the ultrastructure of the extracellular matrix after myocardial infarction. To solve this problem, what we first did was we established a mouse model of myocardial infarction. So in particular, we surgically ligated the left anterior descending artery in mice, which then prevented blood flow to the areas distal to the artery, which would then lead to ischemia, necrosis, and death of the muscle tissue. And the reason why we chose to ligate the left anterior descending artery is that it is one of two main arteries that supplies the left ventricle. And in humans, it is the artery most susceptible to atherosclerosis and hence myocardial infarction. We then took harvested hearts from our mice, and we took short axis sections across the harvested hearts, and you can see the histology in panel 1C, and you can clearly see in the sham-operated mice a very muscular left ventricle and a smaller right ventricle. However, after myocardial infarction, the left ventricle muscle gets replaced by a dense collagen scar indicated by red in these bright field images and in orange in these polarized images. So then we sought to quantify and quantitatively analyze the collagen matrix ultrastructure in our fibrosed hearts. And what we can clearly see under these images is that there's a clear progression in the sort of quality of the collagen matrix ultrastructure. We wanted to quantitatively analyze this. And the way we approached this was we took these images that we took 200 images in total. We took these images and then color deconvoluted them into 3 different channels, and then subsequently binarized and skeletonized these images so that a computer could understand them and read them. And once the computer could understand and read them, we allowed the computer to sort of assess 294 different features of the collagen fibers in these images. So features such as how long the fibers were, how wide they were, and then we were able to plot these 200 images onto a dimensionality reduction plot or a T-SNE plot. And each dot represents an individual image, and the similarity of 2 images is represented by how close 2 given dots are to each other. And what we can clearly see on this T-SNE plot is that the ultrastructure follows a clear pro-fibrotic trajectory from the sham-operated healthy hearts in purple to the post-op day 4 in red, post-op day 7 in green, and post-op day 14 in cyan hearts. So there's a clear pro-fibrotic trajectory from a healthy myocardium to a fibrosed myocardium. So in conclusion, we demonstrate that after myocardial infarction, there's a clear formation and clear maturation of this collagen scar. And then quantitatively, what we see is that there's a clear pro-fibrotic trajectory after myocardial infarction. So what do we think is going on? What we think is that there's a clonal proliferation of individual cardiac fibroblasts. So in panel 1C, what we see are these very short collagen fibers at post-op day 4. And we think that this represents clonal proliferation of individual cardiac fibroblasts. So this is one cardiac fibroblast clone. Here's another cardiac fibroblast clone. And the way we seek to hope to investigate this in the future is by using the rainbow reporter system. And this would allow us to individually label individual cells with a different color. So for instance, one clone will, and all of its progeny will become orange, another clone and all of its progeny will become red, etc. And hopefully by doing so, what we'll see is these expected results in panel 3C, where fibroblasts will proliferate from the border zone of the infarct and into the actual infarct zone. And this is how we hypothesize that the collagen that the collagen scar actually forms after a myocardial infarction. So in summary, in this poster presentation, I've talked about how we established a myocardial infarction model in mice, and then we were able to quantitatively analyze the ultrastructure of the collagen matrix in tissue sections from these mice, and how in the future we hope to investigate the cellular mechanism by which the scar actually forms, which we hypothesize to be due to clonal proliferation of individual cardiac fibroblasts. Thank you for tuning into my poster presentation today, and I hope you have a great rest of your day. Take care. Bye-bye.